Okay. So it's my turn. I really enjoyed your presentation. I think it's different. Yeah, it's really good. So I'll tell you about. Strange, I don't see you guys. I'll tell you about the group and what we're doing. And we're doing single molecule biophysical chemistry. So we're interested in the structural dynamics of what's happening. And we're looking either, now I'll get the pointer here. And we're interested in structures that can fold in a biological environment, also structures that can fold in a non-biological environment that can have some technology applications. And then, because we're interested in the dynamics part, we're have turned to single molecule microscopy and the use of fluorescence. So that's a little bit in a nutshell what we're doing. And I have a picture of the group. And here on the left side are people that are not anymore in the group. And on the right side, people that have joined since the picture. And Alexandra, Sophie, and Maria are in the COFOLD project. Good. So in the way I thought is I would tell you a little bit of what we have been doing before and why we're using fluorescence. And then I'll tell you a little bit about COFOLD. So as I say, we're interested in this structure and dynamics, which I think fits well in the COFOLD theme. Structures are always dynamic. And the time scale, basically, of the dynamics depends on what is moving. So if it's a bond that's moving, then this is vibration, and this is very fast. It's in the femtosecond range. If there is more a secondary structure kind of movement, then this gets longer, and it's this microsecond, millisecond, and can even be even longer type of movement. And it is so that fluorescence, the lifetime is around here in the nanosecond range. We can have pulsed lasers that can have picosecond or femtosecond pulses, so we can probe and start to probe. And then when we're thinking structure and distance change, then we can use this FRET method that looks at distance change, which we can then resolve in time. And then I added that because I think that a single molecule method by itself can bring errors. So I think we always have to do the work and do the ensemble measurements, which are the structure using, for example, CD, or binding using different methods, or looking at the stability of the structure, which also brings important information.
So maybe explain some of the graphs. So the top one is a circular diagonal, where you and the gel. So just for the bioinformaticians, maybe they don't know what these things.
Oh, so this tells us about the secondary structure to some extent. The binding tells us if they're bound or if they're multimers. And then this is a binding curve where we get a KD. And this one is melting experiment, where we get melting temperature, when it tells us something about the stability of the construct. Good. So this single molecule FRET, the way it works is that we have this construct that's labeled with a donor and an acceptor. And here it's immobilized on the surface, so you can maybe see donors and acceptors. And then we follow that as a function of time. here you can see that the donor is changing as a function of time, and the acceptor is changing, and it's anti-correlated because they're coming together and away, right? And this one is just a check. So from that, we can determine a FRET efficiency that tells us exactly that it's changing its conformation. And with this method, we can get time traces for each individual molecule and seeing how they're changing their conformation as a function of time. So if we have our sample that's forming a structure, so it could be unfolded and then sample different folded states. For example, in this trace, where it's getting at the end to the most stable state and staying there some time. And we can plot either the time traces, where we can see the dynamics and reactions pathways, or the plotted as function of histograms, where then we see the conformational heterogeneity. And this one, which is the state that's the longest, is also the one that's most populated, right? That's nice. It's thermodynamics. It's working. Good. So we've set up this method, and we're doing different things, or we have been doing different things. One is to think about how crowding conditions influence this folding process. And is it influencing? So we've been looking at this model structure. That's a DNA structure that folds a G quadruplex, and looking at it with different amounts of synthetic crowding. And it's not a surprise that the most crowded environment gets that the unfolded structure is decreased and the folded structure is increased. This is already known from the literature, and it's just that it gets confined, so it needs to fold. What we saw is that we have the dynamics. So we actually expected that it gets trapped in the confinement and then cannot unfold. And this would be what is driving the equilibrium towards a more folded structure. But what we see is that it is the folding rate that gets increased with the concentration much more than the unfolding rate is affected. And so we explain it by coming from an unfolded state, you need to overcome a barrier towards a folded state. And when we have crowding and we have something that's folded, then this transition state is actually could be a state that's not completely folded, but still much more compact than the unfolded state. And then because of the crowding, both this transition state and the pure folded structure would be affected. And so this would strongly affect the folding rate and not change the unfolding rate, which would be between the folded state and the transition state. Okay.
One question is like, so normally these are driven by falling delta G. So here in the first graph, you kind of end up in a stable state, and then you get finally to a less stable state, or just like, yeah, where the bar is on the graph here. So
no, this one should be lower.
Yeah. Okay. Good.
This one should be lower.
Yeah, and then you just further.
Yeah, okay. It's just to explain, to put the same thing on the whole graph.
Okay.
But you're right. This one should be lower.
You upwards.
Yeah.
So
Absolutely.
Good.
Or maybe it's without salt and it only folds if it's crowding. I don't know. That was what the experiment.
No. Oh, it's with little.
Yeah.
Yeah. Yeah, it's an even better explanation. It's with very little salt that we've done that.
Yes.
So
it's
actually, without
so
crowding,
actually more
it's
stable than
more
the U
the U
state.
state
Yeah.
is more
Okay.
stable.
Okay,
Yeah,
good.
but this doesn't change the message that this is what is moving. And I think it's moving independently of this position.
Yes.
Yeah. You should present. Good. So that was one thing we're thinking about. The times, the rates that I've shown are relatively slow rates, and this we can catch by immobilizing the molecules and looking at them. Some of the folding rates can be quite fast, and we have started looking at folding when we can look at fast times, and then we have molecules diffusing through a confocal spot, and this is work by Alexandra. Then we can see this burst coming in and out, and analyzing that, we can get a very fast times of folding and of intermediates. I wanted to show the work by Emile because it got published recently, where we looked at a longer sequence. Here, so it's the same repeated sequence again and again, so it can form a number of conformations. And this is the Fred histogram. So you can see it's a quite broad Fred histogram in these conditions, but there seem to be two main populations. Something that is high Fred is folded, and something that's a low Fred is not folded. And the nice thing with these single molecule approaches is that we can see dynamics. So we can see that these unfolded states are quite dynamic. And then the folded states, they stay longer in the folded state, but there is also some dynamics associated. And then what we also saw is that this long state here is identified with a fully folded structure. And what we also saw is that if we look at kinetics and see how long it takes to get the fully folded structure, so there's a part that's very fast and then a part that's quite slow. And we came up with a model. This is the unfolded one, and then it can fold many different structures here or these two, and then this one is the most folded and stable one. And we came up with this nice and very complex model to explain our data. And these are too many parameters to fit this curve in any reliable way. But what we found out is that we can extract some of these rates in this model from our time traces, and then putting them inside the model, we get a nice agreement with our data. So this shows that there is, in this case, frustrated folding of these sequences that have many repeated sequences again and again. Okay. So these are for folding. And then using the same method, we can also look at interactions with proteins. And here we have proteins that are labeled. So we can see that this protein has found the DNA and then binds very shortly. And then in the next example, the protein is not labeled, but it finds the DNA here and then starts to slide, process the DNA, and then the DNA is resisting coming up, and so on and so on. So with that, we can study how the protein interacts with the DNA, and during the process, is it blocked by something, or does it just go straight?
So what does the enzyme or protein do again? Does it like unfold or does
This
it...
one unfolds.
Okay, yes. Binds and then
then tries to unfold.
Yeah.
That's why it's going down.
And why do you see? So you label the enzyme and then you label the end of the...
No, so
strange.
this one we label the enzyme and DNA.
So then you only see when it
So
gets
we
to
see
the
binding.
end, or
No, so we just saw binding,
Yeah,
this
binding,
one.
yeah.
And this one we just label the DNA. And we see processing.
So I don't understand the figure down there.
Yes, this one should be for this one. Just to show that there's binding.
Yes.
Hmm?
Oh, so the two orange strands bind to each other and then get the two fluorophores close? Or so I just don't, I don't understand.
Yeah, so
what's going
a
on.
way to look at, so I was telling you about fret, and then fret needs to have a distance between 2 and 10 nanometers, but we can look at binding of things that are much further away, just by looking with co-localization.
Mhm.
And this is what I'm trying to show here. Okay.
And it's co-localization by the two DNA strands binding to each other.
That's just the schematic, right? It
Okay.
doesn't need to be a DNA, it can be a
Oh.
protein or something else. It's just to show that there's binding.
Okay.
And we're also, so this is in vitro, and this is where we have the most control. We're slowly starting to think about cells and how cells are working and doing their stuff, and where some of these enzymes are situated in these cells. So we're also gearing up to do cellular imaging. Good. I also wanted to tell you about this one. And it's work by Alexandra, because she did a really nice work looking at the structural stability, or how these larger DNA origami structures form. And with AFM, looking in conditions where they form nicely, and then when they do not form, here is with magnesium and here is with sodium, and then putting fluorophores on the structures and doing similar experiments with single molecule fret. So here is the structure forms well, and here it does not form so well, it's lower fret. And for some of the projects that we have, we don't want to have magnesium so much. So what we thought is...
But what does magnesium do?
It just helps fold the structure.
Yeah, but does it also, why don't you want it? Is it because it also affects the fluorophores? Or?
No, because it affects the sample we're putting on the DNA origami.
Okay.
Okay. So we were thinking, what happens if we start in magnesium and then turn to sodium? How does it look? So in magnesium, it looks very well. And then when we add sodium, it looks a little bit worse. So you can see that this is in magnesium than in sodium, but still it looks much better than if we start with sodium alone, right? So the way we've prepared the sample really mattered, that with magnesium, even if we washed things, it looks better. And then we thought, well, Alexandra thought, let's start in sodium and add magnesium. And this is in sodium. And then by adding just a tiny bit of magnesium, once the structures are not well formed, then they form again. And actually this recovery is quite big. Good. So this is what I wanted to tell you. And I also wanted to tell you that we have an infrastructure where we have several microscopes that can look at one that can look, it's good at looking at cell, one that's really good at doing this time resolved very fast dynamics, and one that can do higher throughput. And we can look at different aspects. And I know here that we're coming with different methods and interactions, and I'm really convinced that single molecule fluorescence can actually be pushed to interact really well with the computational aspects, and also be pushed to interact with the cryo-EM in a good way. Good. So what are we doing in the COFOLD project? So Alexandra just started today. Sophia is slowly starting up. And Maria, who's not here, is doing a project in the group. She's a master's student. And our role is to look at kinetics of co-transcriptional folding in this wave package 2. And we're having 2 sub-projects. The one is looking at parameters of folding kinetics, and Alexandra will work on that. And then we're very interested to hear about all these nice structures that you found that fold differently, co-transcriptionally or not. Then we can test them here and give input on what the kinetics is. And then in the second sub-project, it's RNA folding studies using fluorescent aptamers. So this is Sophia. And we're learning to use these fluorescent aptamers. Maria is also working with that. And we're interested in how they fold, the kinetics, but also to study fret between these aptamers as tools to tell us more about larger structures. And then we also will be happy to contribute in the work package 3 on the RNA device-based and kinetics of the trapping. I guess that's also via these aptamers.
Yes.
Yes.
And that's possibly, yeah, Nikolai.
That's you.
Yeah.
And on work package 4, where we go in cells, and that's Ayana.
And Adam. Yes.
And Adam. Perfect. That's what I wanted to tell you.